Good afternoon, ma'am. I will report my topic in bam302, and my topic was all about mass production of pathogenic fungi, state of the art. Actually, this is the part 2, ma'am, because the part 1 will be reported by Charles Olasiman Saire, and I have not in contact with him. Here's my topic. The introduction. What is entomopathogenic fungi? Entomopathogenic fungi are a group of fungi living in soil that infects insects by penetrating their cuticle to penetrate their bodies to eventually kill them and feeding on them. Entomopathogenic fungi are parasitic microorganisms with an ability to infect and kill arthropods. They are mainly biopesticides in ecologic farming as a safe alternative to toxic chemical insecticides, although some of them are also used in biotechnological processes or in Chinese medicine. How does entomopathogenic fungi works? Entomopathogenic fungi produce conidia that attach to the insect's cuticle, and an enzymatic production process mainly through fungal protease production takes place. These enzymes, together with a mechanical process through appressoria development, break the insect's cuticle and penetrate the body of the insect to invade and kill. Production methods for important insect pathogen fungi. Lagidium giganteum, the principal species studied for developmental with larval mosquito as its principal target. The Leptolegnia chapmani has under the study for a number of years as an alternative mosquito control agent. Coelomyces spp killing is notable for alternating sexual and asexual stages and involving copepods or astracods as alternate hosts. Entomophorales is the most species within the group, as are insects pathogen, with a few species also attacking nematodes and mites. Microsporidia is an infection process most common involves the host ingesting the spores, explosive spore germinations in the host gut. Ascomycete hyperoceres conidia are the spores that are produced on the exterior of fungus-killed insects. Laginidium giganteum is a key aspect of a fungus that is in the infectious stage is a mothyl zoospore that actively seeks out hosts and is produced by either mycelium or from germinated resting spores. Elution of nutrients from the mycelium and carrier matrix is required for spore production from competent mycelium. The Leptoleginia chapmani, much less work has been done regarding to this L. chapmani, primarily because the fungus remains in the experimental mode. Pilisa et al. Evaluated as a series of agar-based media for zoospore production, although they observed that most media supported mycelial growth equally. Zoospore production in agar medium contained 10% post-strip. Colimosais spp killian. This group of more than 70. This is a group of more than 70 species within the phylum Chitidromycota or Blastocadialias are aquatic obligatory pathogens of aquatic Diptera, including Colchicida, Psychodidae, Chiromunidae, Simolidae, and Tabanidae. Entomoprotorales is a major impediment in developing, even for efficient and utility biocontrol efforts. Much less inadaptive application has been their biology. The Condidora are short-lived after the vivo production and extremely difficult to produce efficiently in vivo vitro production. In vivo, hyphal bodies of protallast are in basic vegetative stages of the spongi resting spores, however, have deemed to be more promising infectious propagule to biocontrol efforts. Under specific environmental conditions, the resting spores germinate to produce primarily conidia, which given to rise infections conidia. Microsporidia is a salient feature of microsporidia is their obligate intercellular development. Thus, these organisms can be mass produced only in vivo in their host or in vertebrate cell culture. For example, locust T is produced by performingly infecting grasshoppers with a dose that is optimal for spore production, allowing the pathogens to multiply within the host. Dayon, asomycete hypocrylase is under certain liquid fermentation conditions, mainly substitution of inorganic or organic nitrogen. It can be produced as myricide conidia, and these conidia are not true conidia and are produced on the ends of hyphal strands. Lastly, mycelium, the last major form of fungal vegetation, proliferation of a mycelial derivative such as the microsteria can be used. Solid substrate fermentation. The end product of solid substrate fermentation yields dried grains kernels colonized by Viveria, in particular Metarsium anispoliae. The process and quality control in mass production. A quality myoinsecticide demands a thoroughly quality assurance competent to any production effort. Despite what ostensibly seems to be fine control of fermentation variables, the production process is only semi-controlled, especially when on a large commercial scale. The second is there's another problem in mass production efforts. Genetic changes in the fungus and expressed the situation succinctly. Entomogeneous fungi will denigrate when continuously cultured on nutrient-rich media. Genetics can change and encompass virulence determinants. It also changes in colony color and gross morphology or decline in conidial or metabolite production. Number 3. Changes in virulence with repeated subculture vary widely.